going to tell you today about a project called DIPY, which stands for Diffusion Imaging in Python. And the method that we use to analyze brain structure, to measure brain structure, is this method called diffusion MRI. We've known for a long time that normal behavior or cognitive abilities are supported by brain connectivity. These are neurologists who in the 19th century noticed that disconnections of certain parts of connections between different parts of the brain, those are associated with particular kinds of neurological disorders or disorders of behavior. So, and they had the insight to say that what is important about these neurological disorders is the disconnection of different parts of the brain. So we understand that connections between different parts of the brain are important, and over the last few decades we've started to understand that the connections through nerve fibers between different parts of the brain are not just passive cables. They change with development, and there are individual differences that account for differences in behavior, even beyond neurological disorders. And these connections actually adapt with learning. This has clinical significance because measuring the properties of these connections between different parts of the brain can help us diagnose different disorders in detail. The method that we use, as I said, is magnetic resonance imaging. There are many ways to measure MRI. Some of them focus more on neural activity, functional MRI. Some of them focus more on anatomy. Those are often referred to as structural MRI. And there are many other methods to measure MRI. Brain connectivity in particular is measured using what we call diffusion MRI. Diffusion MRI relies on the fact that the brain connections here depicted by opening up, actually dissecting out parts of the brain to expose these wires that run through the white matter. These wires are made out of large collections, bundles of individual nerve fibers, individual nerve cell axons that run through the white matter. And diffusion MRI relies on the fact that, let me just load this. Sorry. Diffusion MRI relies on the fact that water inside of these nerve cells diffuses randomly and moves around in this Brownian motion. And in cases in which the cells are just sort of spherical structures, diffusion is equal in all directions. And we call that isotropic diffusion. But if we're looking at one of these nerve bundles, there might be more diffusion along the length of the bundle than across the boundaries of this fiber, and we call that anisotropic diffusion. And we use the application of magnetic field gradients to differentiate between this radial diffusivity and what we call axial diffusivity along the length of these fibers. So here's what the measurement looks like. So here we're looking at a slice on the top right. We see a horizontal slice through the middle of the brain. Down the bottom, this is sort of a coronal slice. So we're seeing left to right. And then at the bottom right, you can see from the back to the front of the brain. And this arrow depicts the application of magnetic field gradients in different directions. And as you can see, as the magnetic field gradient rotates around, the image in different locations brightens and darkens. And that is because in those locations there is anisotropic diffusion. Diffusion is different in different directions. Now, from this measurement, we can infer in every single location measurements on the order of cubes of approximately a millimeter on each side, we can get a rough estimate or a summary estimate of the diffusion in those different locations in all those different directions. So this is what it looks like for a single location in the brain, again, a cube of roughly 1 millimeter on a side. Each point in this point cloud is a measurement in one direction, and the distance from the center of this point cloud is an estimate of the diffusion distance of the water inside of this cube. Now, as you can see, the diffusion is in this particular location is quite anisotropic. The first step usually in analyzing this kind of data is to fit some kind of a model to these data. So one classic model is this model called the diffusion tensor model. This diffusion tensor model estimates a three-dimensional Gaussian distribution of diffusion. And we can summarize this through an ellipsoid that describes this point cloud. So now we have summarized this complicated measurement, in this case in 150 different directions, in just a few parameters that describe the distribution of diffusion in this location. Now, from this model of our local diffusion, we can do a variety of useful things. We can summarize the diffusion in that location, but you can also go from the diffusion in that particular location to tracks that connect different parts of the brain. And this is how we do it. So again, we're looking at a horizontal slice through the brain. If we zoom in on a particular location here, what we can see is we've modeled the diffusion distribution in every one of the voxels, these volumetric pixels. And we can see that we have an ellipsoid in every one of these voxels representing the distribution of diffusion in that location. Now we can propagate a streamline through this, treating these ellipsoids as a vector field. We can propagate a streamline through this and estimate the connection through this part of the brain. Now we can repeat this many, many times, estimating the large-scale structure of the connections through, for example, in this case, the middle of the brain, the large connections through the corpus callosum that connects the two hemispheres. So that's roughly the process of modeling from the signal all the way to the tracks. So in this point, I'd like to just stop and say these are all steps that we do using this software called DIPY, Diffusion MRI in Python. It's part of a larger community of practitioners called NIPY, Neuroimaging in Python, which includes many different projects analyzing different kinds of MRI data mostly, but also other neuroimaging modalities. DIPY was started in 2009 by Eleftherios Garifalidis, who's still the leading developer of this project. And there are contributors from many different places, many different labs that still contribute to the project. And as I said, DIPY implements many different ways of modeling individual voxel data and then applying these tracking methods. There are other methods for image registration and segmentation that I won't be even talking about today. Now, diffusion MRI has a serious problem of validating the measurements and the models that we're making relative to the brain structure. This came off the screen, but says algorithm 1 and algorithm 2. It just marks these two images. Part of the large problem is that there is a large collection of ways to measure diffusion MRI, and there are a large collection of different algorithms that can be applied to analyzing diffusion MRI. And when you use different measurements and different algorithms, you get different estimates of brain connectivity. We have one algorithm, we're looking sort of from the side, inside the brain. At the top, we have an estimate using the diffusion tensor model that I talked about before of two different connections. In silver, the arcuate fasciculus that connects the temporal lobe, areas in the temporal lobe to the frontal lobe. And then in orange, the corticospinal tract, which controls motor connections. In the bottom, we have another kind of model and another kind of tracking algorithm used to model these data. And you can see that these two algorithms produce very different results. They tell us something very different about the biology of these connections. Furthermore, when we look at, in this case, a surface of the brain from the side, the connection map varies quite widely also. So this is the connection map for this arcuate fasciculus. And you can see that the termination endpoints are quite different depending on what algorithm was used. So this poses a large challenge to us. And what I'm going to present here now is a statistical learning approach to this problem, something we also call in vivo validation. Some have proposed that you might take this brain, make a measurement, and then open up the brain, but that poses a problem because I don't think we could find many volunteers to participate in that kind of study. And also, the measurements that are done using anatomical methods, using dissections or using histological staining, those methods have their own problems. Instead, what we're proposing is that you could validate the method through an cross-validation approach. The idea behind cross-validation is that you make two measurements, and you can compare the two measurements to each other to get an estimate of test-retest reliability. So here, for example, we have a measurement, again, 150 different directions in one single location in the brain, one single voxel. And the difference between these is an index of test-retest reliability. Another way to compare these two signals is to first estimate a model on one of these signals and then compare the model predictions to the second measurement. And we call that cross-validation. We can compare then the error in estimating the model to the error in test-retest reliability and calculate an index. We call that relative root mean squared error. That tells us how well the model is doing relative to test reliability. If relative root mean square error is smaller than 1, it means that the model estimate is better than test-retest reliability, and we think of that model as a good model of the signal. Now, we've developed an API to do these kind of cross-validation tests of models in DIPY. This is falling off the screen as well, but I'll just tell you what you see here. The idea is that you define a variable that tells you about the gradients that were measured. That's this gradient table. It's an object that represents the measurement. And then you can define a reconstruction model. So reconstruction of individual voxel signals, we call that. The modeling of individual voxels, we call that reconstruction. And then you use this model to fit an individual instance of the data. That produces what we call a reconstruction fit object. This fit object can then be used to predict a new signal, or the signal based upon this model, on, say, the gradient table that we used before, or another gradient table, another kind of measurement. And then this prediction can be confronted with the data. For example, in this case, we're using the tensor model that we've implemented in DIPY. You fit that to data number 1. You predict, based again on the gradient table, and then calculate a root mean squared error relative to another data measurement. Then you can calculate relative root mean squared error. And this is what a relative root mean squared error map looks like in a brain of the diffusion tensor model. So you can see that in many places, the relative root mean squared error goes towards the green, lower than 1. That means the model is better than test-retest reliability in those locations. But there are particular locations in which relative root mean squared error for this model actually goes above 1. And in one particular location like that is a case in which there are crossings of different nerve fibers around the corticospinal tract meeting with the corpus callosum. And we know that the DTI model is a good model for locations in which there's only one big fiber bundle going through. But for these crossings, we actually need another kind of model that incorporates the notion of crossing fibers. And when we use a model like that, we can see that relative root mean squared error is now pretty much in the entire brain smaller than 1. So this model actually captures the signal very well. And this kind of validation gives us, or this kind of result gives us a validation that this model is a good model of the signal. If you've only measured once and you can't do this cross-validation, we've actually implemented a k-fold cross-validation scheme. There's a function called k-fold cross-validation that takes an individual model object, data, and the number of folds. In this case, we just split up the data into 2. And what it does is it splits the data up automatically, fits to half, and then compares to the other half, and vice versa, creating an entire prediction with for directions that haven't been put into the measurement. So it's entirely cross-validated. Now, you can put in another model. In this case, the CSD model is a model that incorporates crossing fibers. The API for calling that is exactly the same. And the important design aspect of that is that each one of these models by itself has a predict method that can be used so that this k-fold cross-validation function doesn't even need to know anything about how a model predicts, as long as it knows that the model can predict given a new gradient table, a new measurement scheme. And this allows us then to incorporate additional models into the same function. Now, this idea, this notion of in vivo validation, of checking the predictions of models against the data, applies also to the larger scale measurement, the larger scale models. So these tracks are also, in a sense, models of the signal. And while I was a postdoc at Stanford, Franco, Pestilli, and myself and others in Brian Wendell's lab, we developed a method called LIFE, or linear fascicle evaluation, that allows you to do cross-validation at the level of the entire fascicle. So previously I was talking about cross-validation of individual voxel signals. In this case, we construct a forward model from the tracks to the measured signal to estimate the match of the signal as predicted from these tracks with the actual measured signal. The idea is that instead of going from diffusion to tracks, we now go from tracks to diffusion. So each one of these streamlines has a forward model predicting a signal in some voxels. In the voxels in which this track doesn't pass, we don't predict anything. That's that straight line. Maybe another track might predict a different signal in a collection of voxels, and we get a large matrix like that for all the tracks that we estimate. And we sometimes estimate on the order of 500,000 tracks in an individual brain. So this matrix goes on for quite a bit, both in that direction and in that direction. We then compare that to the actual measured signal. Now, this scheme forms a linear model. So the matrix of the signals predicted for each one of the fibers across all the fibers is on the left side here as the big design matrix X. Y is then the signal measured across all the voxels. And now we need to estimate these betas, the weights on each one of the tracks, to tell us which one of the tracks actually fits the signal. We solved this big matrix problem for non-negative weights, where part of the challenge is that this ends up being quite a big problem. This also has a similar, in DIPY, also a similar API where, again, the model is defined with the measurement. It's then fit directly to the data, but together with the tracks in this case. And then, again, we can predict. This kind of API also has the additional benefit that we can then find the tracks for which these betas are larger than 0 and filter the tracks, so reduce the data size quite substantially. Okay, to summarize, we talked today about measuring brain connectivity with diffusion MRI, specifically how we use the DIPY project to estimate the connectivity between different parts of the brain. I've told you about the validation problem that we have in diffusion MRI and the notion of in vivo validation through methods taken from statistical learning and how we've applied this in DIPY. Finally, I'd just like to mention collaborators on this work in DIPY, especially Eleftherios Garifalidis, but also Stefan van der Walt and Bago Amirbekian, who were involved in some of this statistical validation work. There are many other contributors who I don't mention here. And also my collaborators in Stanford, Brian Wendell, who was my postdoc mentor, and Franco Pestilli, who did much of the work on life. And I'd be happy to take questions if I have time for that.
We have time for a couple of questions.
Yes.
So it looks like what you've got there is with the model track, with the tracks, it's almost like a kind of a nonlinear dimension reduction, right? And in a way, the kind of validation back to the model, so you just kind of like kind of like some of the drugs that are with deep learning, the encode decode thing. It occurs to me, would there be any benefit in actually doing just a non-model based dimension reduction and then doing some kind of analysis at the level of what's inferred using something just like regular ICA or something like that versus the tracks and seeing if there are correlations there?
Yeah, the problem there is the spatial continuity that needs to be maintained, right? So if you take the signal, the raw signal, and you decompose it, you might decompose, for example, you might decompose together different parts of the brain that are sort of pointing in the same direction, but are not spatially contiguous. The real challenge is maintaining the spatial contiguity in this kind of scheme, which is hard. And that's why we start, bottom up and then go top down from this top bottom up.
But I would view it more as an enhancement to the cross-validation approach, so that you could actually see how the common sort of factors or whatever that you get from the numerical
I'd have to think about that more.
Okay.
Sounds interesting.
We have time for one more question.
So I'm wondering whether or not, I'm looking at the models that you're implementing, and it looks like they're mostly based on some DTI. Do you also incorporate like hard use type applications?
Yeah, so I didn't have much time to go into this, and DTI is kind of a classic example, but we have implementations of many different kinds of models, including hardy models, diffusion spectrum imaging, Q-ball, everything from Q-ball, CSV, QBI. There are many different reconstruction models there. Yeah, the general philosophy of the project is to implement many different models because different models